Welcome to the Clinical Research Podcast, bringing you the latest developments in research explained by our world-leading clinicians, academics and scientists based in Nottingham. Clinical research is very much about collaboration, which means it can often also be very much about lots of organisations with lots of initials. We try not to burrow too deeply into that particular rabbit hole unless we really have to, but for this episode, we really have to. Rather than people or particular studies, it's about plans for the future of clinical research in Nottingham, which means understanding a little bit of how all the parts fit together. A lot of health research in the UK is planned and funded by the appropriately named National Institute for Health Research, often abbreviated to NIHR. One of the ways the NIHR organises research is through a network of biomedical research centres, often abbreviated to BRCs. Those BRCs in turn bring together hospital and university researchers to focus on how academic research can be translated into practical results for patients. And Nottingham has one of those BRCs, known unsurprisingly as, putting all the parts together, the NIHR Nottingham Biomedical Research Centre. Like most BRCs, the NIHR Nottingham BRC focuses on a number of different diseases and conditions, and we group the studies for these together in 6 research themes. The themes involve respiratory medicine, hearing, musculoskeletal, mental health and technology, and gastrointestinal and liver disorders. In Nottingham, the 6th theme provides access to our expertise in magnetic resonance imaging, and as you will hear in this week's episode, this is central to the research being done by the team from Gastrointestinal and Liver. You'll hear 4 people. In order, Professor Guru Aithal, who leads the theme, then Professor Penny Gowland, who's part of the magnetic resonance imaging team, then Neil Guha, who's a hepatology professor, and Dr Gordon Moran, who's an associate professor of gastroenterology. A couple of the team mentioned some work by a colleague called Luca. That's Professor Luca Marciani, whose study, called MAGIC, helped treat constipation in children by using markers to track food moving through their digestive systems. There's more information on all of them in the show notes. Professor Aithal started by talking about how funding came to be focused in specific research areas.
We had 3 main areas, which were infections, which is H. pylori, Clostridium difficile, and viral hepatitis. Then physiology, which was relating to inflammation-related consequences of body physiology, as well as liver metabolism-related body physiology changes. And we were using imaging as a tool, as well as a test.
From my point of view, we were always welcomed within this theme, and the idea of doing interdisciplinary science is that everybody brings their own expertise to the table. So we brought the imaging, and we could work with people who had interesting questions and were able to express them to us in a way that we could then design the answers. I've been meeting with Gordon this morning, and you have to say to the people who've got the problems, what exactly is it you're looking for, and then we can design the solution to fit that. Whereas, just us coming up with a method is no good. We have to be working absolutely in lockstep. We're part of the group, even though we're not formally part of the group.
Walk me through, have you got an example, maybe a research question that you guys wanted to deal with and how the imaging was able to help with that?
When I first was appointed in 2009, I was interested in finding better tests for not only design for understanding the severity of liver disease, but how it affects outcomes. So I had a meeting within 6 months of starting with Penny and Sue Francis about some of the imaging protocols that they'd already developed for liver disease. And we set about designing a study where we got a new imaging protocol that not just looked at the liver, but looked at the heart, the kidneys, and the blood vessels in a cohort of patients with severe disease. And we tracked them going forward. And we were able to track them so we could predict which patients were to go and get a clinical outcome. And that was novel in terms of bringing together all those protocols from using our MR scientists, from Penny and Sue in the group, It was also novel in terms of it was the first imaging study to look at those outcomes together, and it used the infrastructure in terms of our large burden of liver disease. And that study was then published 4 years later, in one of our leading liver journals. For me, that was something that we were only able to do because of the BRC. I don't think we'd have been able to do that in other centres.
That's quite an interesting contrast, because what Neil's just discussed is the need for the entire infrastructure to make that work. You know, you need the cohorts of patients, you need them monitored, you need them tracked, you need to bring them in. The whole of the bigger BRC infrastructure is absolutely essential to that. The imaging, in that case, I guess in some ways we knew what we wanted to do there. The measures were, at least the ones that were done originally, were in the literature. We had to create them, and we had to create them in a way that could be done efficiently and rapidly in a clinically appropriate manner. But we weren't sort of starting from fresh within a sort of completely, you know, what are we going to measure here, which is sometimes more the case in what we do more recently, including in the liver. The example I was thinking of this morning is completely opposite of that. Gordon's interested in the vascularization of the bowel wall, and we don't know how to do it. You know, we don't know what to do. So what we're doing is we're trying lots of different things that we can measure to try and achieve that. And what the question came up to Gordon is, what exactly is it you're talking about? What exactly is it you're looking at? So that we can, because we've now got something, and now we can try and match our outcome measure, our quantification of the data to that.
What we're describing is something very important and unique. So for a study of any form that involves patients, for it to be successful, you need a really good outcome measure. You need something that can be measured. You can repeat it, it's stable, it changes with disease, it changes with treatment. And that's what we were talking today with Penny was, so my interest is in measuring disease activity and scarring as well in the bowel. And what we wanted to measure is, so usually we tend to give an injection called gadolinium, for instance, in MRI, which we try and avoid using because of cost and adverse events and all that. So in this case, we wanted to measure blood supply to the gut, which increases with inflammation, with something called time of flight. But me as a clinician, I don't understand what time of flight is, but actually when we start talking, what time of flight is what I see in endoscopy, which is essentially what we call loss of vascular pattern. Once the blood vessels start getting engorged, you start seeing them. And what time of flight measures that. So then we try to match what Penny can measure in physics to what I want to measure in the patient in clinic. Create something, measure it against the gold standard, and put it out there. And that is, if you like, what we I think Nottingham are good at, because of the skills we have in physics and the clinical cohorts we have, is creating outcomes eventually. So we've got now novel outcomes in liver. We've got, for instance, Guru has worked a lot to try and validate T2, which is a measure in the gut. It was for liver, but it really helps me in the gut as well. We're trying to improve things in fibrosis. But if we can actually create all these measures, this is what academia and industry will use to measure their drugs. So this will bring back the focus to Nottingham as a unit that actually creates outcomes through imaging.
We as a BRC, we as a group have been quite effective in translating discovery into a diagnostic tools. In liver, we have contributed substantially to new way of identifying fibrosis with both blood tests as well as imaging. And you heard some of the MRI tools we have developed from Neil. We can measure portal pressure with that. We can measure scarring in the liver, and luminal gastroenterology wise, I think Penny can tell number of methods they have used have been a contribution from Nottingham.
The most widely exploited tool probably that we've developed is the one for measuring small bowel water. So we can measure the sort of flow through the gut, movement through the gut, but small bowel water was probably the one that nobody else would have thought of, that there's the sort of, you know, pushed the limits of MRI really at the time when we did it, and we did it very early on actually. Small bowel water is not a quantitative measure so much as an index of the small bowel water, but it turned out to be extremely powerful, looking both response to drugs and looking at normal physiology and response to feeding, response to fat. So that was one of them. The other measures that we've come up with are measurement of flow through the guts, measurement of volumes, movement through the gut from the stomach to the colon. But the other thing that's most likely to be picked up clinically, I think, is Luca's beads. So Luca has developed a method using little beads that you swallow and you simply go through your gut. And this allows you to monitor the transit of the food through the gut, whereas normally with MRI, we're looking at a sort of snapshot, and we can't say if this bit of the contents of your gut has been there 2 hours or 10 minutes, or at least not very easily. But with Luca's markers, we can monitor the movement through the gut completely. And so he can then look at the changes in the rate of transit in constipation, or again, potentially in response to feeding going forward. So those two measures are probably the ones that are the most widely used, but there's a multitude of measures. We're currently aiming at measuring pH in the gut, measuring the flow rates, the local flow rates in the gut, the mixing. The thing I'm really keen to exploit that we've never really exploited is the sort of shear rates, the rate at which food's breaking down in the gut. A bit like your teeth grind it up, your stomach grinds it up, your colon, your small bowel causes emulsification of fats. So lots of different measures.
Those kind of diagnostics, what sort of broad uses can you see them being applied to do ultimately?
I would say that, Scarred Liver Project has been one of the outputs from the first BRC. We had, I would say, 3 lines in our first BRC application, 2007 and 8. And that has now changed way the people are identified with chronic liver disease in the community, and suitable appropriate patients are referred to the secondary care for a further test in a cost-effective way, in a patient-friendly way, and in a way where patients don't have to come to hospital for all the time. In the same way, we want to change the way the liver cirrhosis is really also monitored and identify appropriate patients for intervention, looking at more sophisticated MRI measures, where we can measure the pressure building up in the circulation of the liver, where you can look at how the liver problem affects multiple organs. Coming to the test which Penny mentioned, abdominal pain is very common problem in children, and most common reason for it to constipation. But there is no way other than doing an X-ray for children, which is not a necessary exposure to radiation, very crude way of diagnosing or even telling why there is a problem. So this will transform actually pediatric practice when this capsules which Luca is making becomes available.
One of the satisfying things for me is being able to put onto a solid physical basis some of these apparently psychosomatic conditions, or I'm not sure if that's quite the appropriate word, but potentially psychosomatic conditions where it's unclear where the origin of clinical symptoms. And if we can give people a physical understanding of their condition or not, that's important, I think.
One of the other areas I wanted to get onto was patient involvement as well. Are there any particular areas or initiatives that people have been involved in that you highlight there?
It's been remarkable for me to see how that's sort of grown over the last decade. To be quite fair, we were relatively rudimentary when we started. We got 5 or 6 patients with different conditions to sit in a room and look at our patient information sheets, and to change the sort of wording on them. And over the last 10 years, I think we've involved patients in a much more meaningful way. And that's related to the initiatives of NIHR, but also our own local initiatives. And we've been fortunate enough to have some really good dedicated PPI officers, and one in particular, Andy Ragg, was instrumental in getting patients not just to do what I call the sort of, the tick box exercises or the window dressing around that, but actually to sit there and create or contribute to research ideas, to help design the studies, to sit onto trial groups. And we've expanded enormously from that from the early days to now, where we've got a patient advisory group of around 60 to 70 people. And that's now fed into the much larger biomedical research center. And the influence of that PPI group has not just been in Nottingham, but it's had a national contribution. So people are aware of the James Lind Alliance and the JLA, and the Nottingham PAG group really fed into two major schemes of that, including non-alcoholic fatty liver disease and then one of the alcohol initiatives to actually sit there and help do the priority setting. And I know Gordon's had some very constructive relationships with the PPI, so I'd be interested in his thoughts as well, really.
Last night, I was on the phone with Guru at 6, and I told him I need to hang up because I have a meeting. And in fact, the meeting was it was the first focus group meeting, because again, back to outcomes, we want to develop a work stream to try and investigate what good drugs are to treat a condition called proctitis. Proctitis is inflammation of the lower end of the colon. But the problem with proctitis, it's always excluded from trials because industry has no interest, and it's very hard to measure symptoms because there's no measures, because the measures have been born to measure inflammation in the whole colon, not the last few inches. So the first thing we needed was get a bunch of clinicians, but as well a bunch of patients together and saying, okay, fine, is this important? How is it important? What are the treatment options? And this was the first step in what we call a Delphi process, to try and define and quantify the problem, which is then going back to the NIHR saying, hello, this is a problem. The NIHR hasn't seen this yet, but we're working to highlight the problem through PPI, through what we call the NIHR bioresource, which is a huge repository of patients that Nottingham heavily contributes to. And again, it shows how, if you like, wide and cross-fertilizing the NIHR is. I have access to patients in Southampton and Cambridge who all have a condition which Nottingham itself cannot answer, but because of the expertise we bring in, we can answer that question. But if you like, this is the reverse of it, not just creating an outcome measure, but actually using patients, working with patients to highlight a problem and focus a problem, create the solution for the problem. And that's pretty much work from left to right, really. So working with patients throughout.
I just want to highlight that in the last few years, with Luca's project, we have developed a pediatric group to give us feedback on how they feel, how they want to really design studies. And I have started developing more ethnic minorities involvement and try and make sure that their views are heard. Ethnic specific issues are incorporated into our study designs, suitable patient public advisory people are involved in it. So it's important to really show that we are growing in that as well over a period of time.
So aligned to PPI, I think another important aspect is the public engagement with science. And I think we've seen that really in COVID, of how the public have wanted to be educated about the science of disease. And I think we've had a strong reputation in Nottingham doing that. We used to have an annual event where we used to get members of the public to come around all the science stations, and see how the cameras work, see how physics principles work, see how the culture mediums work. And we've also engaged with local schools. On a national level, Penny, Guru and I had the privilege to set up an exhibition at the Royal Society Summer Science Exhibition in 2013. And we were the first NIHR organization to present at that. And we were able to sort of engage with the public, almost, I think 1,000 people came through that stand over a week. We had patient stories, we had an MRI app, we had a spongy liver to show what scarring is about, and 10,000 people visited the website, and we got sort of national media and radio sort of media coverage from that.
Before I move on to the kind of looking to the future bit of this, are there any other bits of current things that are going on that anybody wants to flag up?
The sort of spectroscopy experiments, which are slightly different, they are possibly more basic science, understanding the biochemistry and physiology, which would hopefully allow us to develop future markers which would be clinically valuable. But at this point, they are in the very early stage. They're big, heroic experiments that, you know, cost a lot of money and take a lot of time. But then hopefully they will provide either better understanding of the diseases and the therapies, or new markers. And I think we're probably in the former stage at the moment. So it's more likely to be a better understanding of the drugs themselves, or the treatments, or the conditions, rather than the markers, but they can lead to markers.
On the basis that they're big, expensive ones that take up a lot of time, Penny, do you want to describe them a bit so you know what's going on?
There are some that involve people drinking or eating something has been labelled with a different isotope. So it's not radioactive, but it's got a different NMR property, and so we can then follow that isotope into the body and watch how it's chemically metabolized in the body. But producing an isotope labeled drink, so means, say, growing carrots in special sorts of CO2, is quite difficult and expensive. So they can be, you know, thousands of pounds worth of drinks. You really don't want to knock them over. And then they're going to be followed over several hours. Many of our experiments take several hours because we're studying the physiological process. And then they'll be coming back several times, maybe over several weeks. And then they'll be taking blood samples. You're not just doing the MRI, you're taking multiple different measures. And this applies to a lot of these studies. We're taking blood samples, we're taking cardiovascular measurements, and then all that data has to be processed, and some of it requires other people to do other processing using other analytical techniques, and then all brought back together and put into physiological models. So it's complicated for the patients or volunteers. It's complicated for a whole range of different scientists. It involves nurses, and lots of different specialities to bring the data together to get the final result. And one thing I think we haven't mentioned as we've gone along, I heard a number of people sort of hesitate when they went to say names, is that none of this is the four of us, obviously. There's lots of different people involved.
Yeah, I would say critical mass means that we are just four of us talking on behalf of about 50 people who are in different seniority of the clinical and academic careers. It includes, currently we are supporting in some form or shape about 100 PhD students or postgraduate students, some scientists, some clinicians. We have literally 50 investigators of different grades who all contribute to this.
And these were a spectrum of non-clinical people, as Penny mentioned, engineers who you would never think would be working on Crohn's disease. I've got, in fact, a PhD student who is actually a gym trainer, but he works on spectroscopy investigating fatigue, and he's really perfect because he really makes patients do exercise in a magnet. So he's perfect for this. I wouldn't do it. I couldn't do it. But and we've got clinicians as well.
So I guess this is back to you, Guru. What are the kind of three big things that we're looking forward to over the next few years with any luck?
By 2035, over 2/3 of the population will have more than one health condition. Our focus is taking gastroenterology, hepatology, and pancreatology as our area of expertise. How do we identify these clusters where the organ interacts with other organ in a mechanistic way? We will also look at what the new tests we need to identify these group of people who will get in the future multiple morbidity and how to monitor them, and also use food as a way of intervening with these. How does food modulate the biology of the gut and metabolism of the liver, and how it can really target multiple health conditions in a way, as well as how advanced technology help us to make a step change in both diagnosis and interventions.
You also mentioned what can be bracketed under infection.
Infectious are more common and have more consequence in people with comorbidity. COVID-19 has brought it forward, but our expertise also is in Clostridium difficile. It occurs in elderly people who are made vulnerable by chronic conditions which they have accumulated over the years. So they need to be dealt with so that their infection is treated in a way that it doesn't really make their physical state much worse than better by the end of we have got rid of it. So these are specific challenges we are addressing as well.
We also talked about looking at physiology and metabolism. So is that something that maybe Neil and Gordon want to speak to?
Talking about metabolism, I think, for instance, one of the very underplayed and very under-researched area of inflammatory bowel disease is specifically metabolism and food intake. And what some of the work we've done through the BRC and through other matched funding is looking at investigating. So that historically patients with Crohn's have always been labeled as under-eating. And actually our work has shown that actually they don't under-eat, they equally eat, just that they just eat differently. And some of the work we've shown now is that they eat less protein, for instance. And the way they handle protein is completely different. And the way their muscles handle protein is completely different. So can we optimize protein intake to help with patients with Crohn's disease? And we know that these patients lose muscle and are fatigued. So can we use the techniques we have? We've involved MRI, involved spectroscopy, but as well as involve other colleagues that have skills in food intake studies and acute feeding studies, where we can track protein intake to try and develop this as an intervention for patients with Crohn's, with liver disease, who lose muscle, who we can reverse through protein intake or exercise. So I think there's a number of cross-fertilizing projects that we have the skill through patients and colleagues to actually answer. But that's, for instance, one of the projects we want to do about metabolism and, in this case, food intake.
I'm quite interested in how multimorbidity might actually change how we think and manage disease, and how, for example, it aligns with the changing infrastructure of the NHS. So to give you a much more specific example, one of the things that we're thinking about as the NHS evolves is taking care out from secondary care and thinking much more about it in a community basis. And people have talked about community diagnostic hubs and things like that. Well, if you take Guru's understanding of multimorbidity and you start to put some of those diseases together in clusters, actually, it might be more logical to look after those and diagnose those in those clusters in a community setting. So we can diagnose, stratify those diseases, give patients a prognosis, and offer new treatments. And therefore, that to me is quite an exciting way of how our BRC will evolve scientifically, but also in parallel with the way the NHS hopes to go.
It's a system biology approach you need to really apply to it, where you can investigate multiple pathways or interrogate multiple pathways in one go at one time, and come up with the gist of it in one way, and then narrow it down. So that includes some technology like metabolomics, proteomics, genomics. So Neil's original work was involving developing diagnostics in that way. So that is where Neil will advise us.
As well, I think something else Neil brings in is biomarker development, which I think we all tend to use this word very loosely, and And I think, so specifically, for instance, in inflammatory bowel disease, if we can actually manage to measure fibrosis, scarring, that will be a huge game changer. Of course, then we can actually start deciding fibrosis is not reversible, who needs surgery, who does not. And industry is not very good at, has not yet developed anti-fibrotic drugs in Crohn's disease because we cannot measure fibrosis. So using Neil's expertise in biomarker development to actually create a very refined biomarker, and Penny's expertise at measuring this through imaging, I think we've got two very good colleagues with me perhaps bringing the idea and the patients work together to develop the future. So I think back to where we started.
So just to kind of pick up on that biomarkers and the imaging idea, Penny, what would you say you're kind of looking to in the future over the next few years?
It's two sides of around a seesaw. So one side is making things clinically robust and usable. So we have studies that last 7 hours. We have measurements in liver patients that take 2 hours and at one time point. We need to distill our measurements into the key time points, the key measures that are clinically sensitive, and that's one area of work. And on the other side, it's finding new markers, and I think that means digging down into the biochemistry and the physiology further. So my personal interest at the moment is on spectroscopy and what we call CESS, which is another type of CESS spectroscopy. So trying to get the sort of biochemical information out of the tissue, or at least some degree of chemical index out of the tissue. But I think that we have to always avoid the physicists going off and playing games and coming up with new techniques. They've always got to be grounded in making them useful and keep pushing it, making things quick and cost effective and clinically helpful to patients. So both of those.
Kind of, I guess, to wrap up, and that's probably going to come back to you, Guru. If we look, I don't know, 4 years, 5 years down the line, where would you want to see the GI research in Nottingham? What would you expect it to be?
Yeah, we would start seeing some interventions coming to clinical practice. So like I mentioned, we are excellent at two ends, which Penny highlighted. I would say the depth, which is absolutely doing non-invasively, the cellular science, measuring how the cell produces ATP, for example, to how we can really bring it to a clinical practice and make a test out of it. And we have got that covered. We have got the science there, and we have got expertise and a process as well. What I want to see now is an intervention. So how we can change it now. And I think industry always has a strong presence in this area because they can invest in so much faster. But what we want to do is find repurposed drugs, which are much more helpful in a multimorbidity state. You don't need to kind of find a new drug to target fibrosis in the liver, as well as in the lung, as well as in the inflammatory bowel disease, saying, well, what's a common underlying principle? What we have, we can use it very creatively, cost effectively to solve it. Same way, I would say that many of the antecedent events or precursor for it is lifestyle, diet. So we want to modulate function of the gut, and through that function of the body, using modification of the food that we eat, much more practical, easy to implement. So again, food science is one of the strengths of Nottingham, and we've got expertise in the lumen as well as in the liver. So we want to make the most of those. I would like to see some interventions which we deliver in the next 5 years.
Well, thank you very much, everybody. I think that's just about it. That's brilliant.
I was hoping to pick on Neil more, but we'll try again next time. Pre-submission.
Thank you very much, guys.
All right, that's brilliant. Thank you very much. Cheers.
That has got something. Yeah, thanks.
There are links in the show notes for more information on clinical research in Nottingham, and the website is nottinghambrc.nihr.ac.uk. Our email and social media links are there too. If you want to stay up to date with the Clinical Research Podcast, you can find us on iTunes, Spotify, Google, wherever you normally get your podcasts. The more shows are rated and reviewed, the more search engines like them, and the easier it is for people to find us. So if you can subscribe and rate and review us, you'll be doing it for science, not just for our egos.